I'm going to talk about probably something a bit of a different strand to what James has been talking about. My PhD kind of examined a different area of exercise physiology and health, and predominantly focused on looking at the influence of exercise on bone health in athletes. So I'm going to talk to you a little bit about some of the experiments that I conducted and some of the results we found, and try to get down to the answer of whether there is such thing as exercise-induced bone loss. So exercise and bone research kind of has a long history. So like over the last 50 years or so, there's been a progressive increase in the number of articles published in this area. A lot of it kind of stems from the development of technologies that allow us to observe changes in bone. So in the 1990s, there was the development of what's called dual X-ray absorptiometry. And this device allows us to measure the density of bone. And it's probably what most people would have heard of before, with athletes being tested for like body composition, or if you've attended an osteoporosis clinic, you might have seen that these devices are used to measure bone density. But as this kind of became commercially available in the early 2000s, this is kind of where we see a huge ramp up in the number of published articles related to exercise and bone. And this number just keeps getting larger. And what we've established so far about bone health and exercise is kind of two strands of the effects that it has. So if I just show you this image here, currently our understanding of exercise and bone health leads to two kind of main hypotheses. Generally, it's believed that exercise is beneficial for bone health, such that it improves the density of bone and reduces the risk of stress fracture. However, it is the type of exercise that is most important that leads to this adaptation. So predominantly it's load-bearing, high-impact, multi-directional exercise. So whether that's like things like resistance training or team sports, anything that is going to cause a heavy load that's transposed through the bone allows for an adaptation that's going to be beneficial. Now, bone stops growing when we kind of reach our early 20s. And so a lot of interventions are focused on younger athletes trying to improve their bone density. However, the caveat to this is that endurance exercise doesn't tend to have a significant load associated with it. It's a repetitive type of exercise that poses a metabolic challenge. And what has been found is that endurance athletes tend to have poorer bone mass compared to other athletic populations, but also compared to healthy controls. So there seems to be something about endurance exercise that actively causes a change, or it actively causes a decrease in the bone density, which can result in increased risks of stress fracture. So currently, there's one working theory about what this is. There are many associated risk factors for decrease in bone density, things like nutrition, menstrual health, but there's actually seems to be an active mechanism of exercise that causes a breakdown of bone. And that's largely what my PhD was investigating. It was investigating this acute mechanism that causes this structural change in bone. Now, just to kind of give you a brief overview of what that mechanism looks like, I'm going to put up a slide here of, hopefully, of probably the best understanding of what causes this breakdown in bone. Now, on the right-hand panel there, we've got three figures that were in response to an acute exercise bout over 60 minutes. We can see that there is a change. I don't know if you can see my mouse. I hope you can. There's been a change in the amount of ionized calcium. Now, ionized calcium is what's considered to be the active form of calcium in the body. And throughout exercise, what we tend to see is that during normal exercising conditions, there is a decrease in this ionized calcium. Now, it's not a drastic change. Ionized calcium is often referred to as one of nature's physiological constants because it's maintained in such a narrow range, usually within 5% of its baseline value. But changes of 2% and such have been shown to increase parathyroid hormone, up to 100 to 200% above baseline levels. That increase in parathyroid hormone has been associated with increases in a marker of bone breakdown. So this marker of serum CTX is essentially a collagen marker. As bone gets broken down, gets released into the blood, and we can see what's happening at the site of the bone cells in response to different stimuli. Now, in this study by Wendy Court and colleagues, what they decided to do is go, well, if we think that this decrease in calcium is causing this increase in parathyroid hormone, then if we stop the breakdown or the decrease in ionized calcium with infusing calcium into the bloodstream, does that negate this increase in parathyroid hormone? And subsequently it did. So what we can see is that as they were given calcium, the amount of parathyroid hormone did not really change in response to the exercise anymore. And that further went down to the bone, where we saw no real change in the amount of bone being broken down. Now, this is the best theory we have of what causes exercise or what causes a change in the structural components of bone in response to endurance types of exercise. My PhD largely examined what is the mechanism for the decrease in ionized calcium. What is it about exercise that causes us to have a reduction in calcium and a subsequent increase in the amount of bone being resorbed? There we go. So that was my PhD largely summed up. What I did to try and answer that was I came up with a hypothesis of why calcium might change in response to exercise. Up until this point, most literature had suggested that calcium is lost through sweat, but it didn't seem likely to me that sweat loss of calcium could account for this small decrease and subsequent increase in bone breakdown. What seemed to make more sense to me at the time was that calcium is present in many different forms inside the body. The active form is often bound to different proteins and carried around in an inactive state. But what changes that composition between the active and inactive forms of calcium is the acidity of the blood. So in an instance where we have a normal blood pH, what we'll have is that hydrogen ions will be relatively high, and they will bind onto albumin and calcium be released. But in a period where we have acidity, what we'll see is that hydrogen ions will increase, calcium be released, and then when we have alkalinity, hydrogen ions will decrease and ionized calcium will decrease as well, because it frees up these binding positions on this albumin protein. So it goes from being active to inactive, and then we might see an activation of parathyroid hormone in the system. So I investigated this physiochemical mechanism using a series of 3 studies. First one was investigating the effects of different types of exercise intensity, whether it be below a metabolic threshold or above a metabolic threshold. Each of these elicit a different kind of threat to acid-base balance. The other one was a hypoxic exercise stimulus, such that when you're in altitude, you tend to produce a physiological response that's called respiratory alkalosis. And this is where we get an increase in pH. And then finally, I examined the use of a popular ergogenic aid in cycling and endurance sports, sodium bicarbonate, which increases the pH of blood as well. Now, altogether, these stress this acid-base system and should change the way the concentrations of calcium in the body. Now, I've got a lot of results, and I've tried to filter it down to a few of the main ones here. But what we found that was really interesting was that calcium has this biphasic response, such that during the onset of exercise, what we tend to see calcium do is increase before it decreases. So this actually suggests that the initial acid-base threat of exercise that causes acidosis increases the amount of free active calcium in the system. But as exercise kind of reaches a steady state, usually after about 10 minutes, we then start to get into a metabolic steady state situation where we can buffer any acidity that's in the blood. That then leads to a progressive reduction in the amount of ionized calcium in the system. Now, because these intensities were different and caused different acid-base threats, what we notice is that the pH that is associated with each of these intensities actually explained the variance between the differences. Now, what was more interesting to me was that the PTH response essentially mirrored the ionized calcium response. So rather than increasing at the onset of exercise, we see it decrease before then increasing later on in exercise. Now, one of the most notable findings of this study was that as you stop exercising, we actually see that calcium continues to decrease and parathyroid hormone increases dramatically. This led to kind of the conclusion for me that there is two things going on here in terms of the regulation of calcium. Firstly, one is that calcium is negatively correlated with pH, and that the intensity differences in calcium can be explained by changes in pH, such that if we have a large decrease, we see more calcium being released from those proteins, and if we have subsequently a small increase in pH, we see that the pH or calcium decreases in line with that. Now, if we control for this relationship of pH versus calcium, what was interesting is that the change in ionized calcium when controlled for pH tend to be fairly similar across the conditions, except for the point at which people stopped exercising. So for the purple line here, this is where people reached fatigue, and we saw a drastic decrease in ionized calcium. And similarly, at this point here, this was the last time point of exercise. And when that exercise stopped, we see calcium continue to decrease. Now, this is interesting because it suggests that not only is pH responsible for this relationship, but there seems to be something about the locomotion of exercise, actual muscle contraction, that causes a change in the calcium in the system. So my final study, I essentially put this to the test, and I had people complete kind of interval based exercise with and without supplementation. So I used the supplementation to try and account for the variance that the acid-base changes could explain. And I used the interval exercise to try and see whether stopping and starting exercise actually causes a peak and trough effect in the ionized calcium in circulation. Because if it does, it suggests that some of the change in ionized calcium during exercise is actually the result of muscle efflux of calcium. So what the study demonstrated was that as you take sodium bicarbonate, you get a significant decrease in ionized calcium. And this is what we expect because it increases the pH. And then subsequently through the exercise trials, we get this peak and trough effect in both conditions. Now, I've kind of amalgamated these results to show this a little bit more clearly. There we go. So what we've got here is that they start the sprint exercise. We see calcium being pushed out of the muscle, and then they rested, and we see calcium being taken up by the muscle, and then they sprint again, comes out of the muscle, goes back in, comes out, goes back in, comes out, goes back in. So this suggests that there's part of exercise that inherently causes a change in the calcium dynamics, and subsequently would result in changes in bone resorption and bone breakdown. So the main kind of messages I found during my PhD was that the intensity difference in calcium and PTH are acid-base regulated. So anything that causes a different kind of height or trough in calcium is a result of pH, but the temporal change that occurs over the duration of exercise seems to be related to muscle contraction. Now in future, what I would like to do is try and examine what the dose-response relationship is with duration. So if we extend this from 30 minutes to 60 minutes to 2 hours to 3 hours, how much of a decrease in calcium can we get? And then I'd like to see how we can try and mediate that decrease. So using things such as calcium supplementation to minimize that disruption. Because whilst this is very mechanistic research in essence, the impact of this is that should someone go through this repeated calcium decrease in response to exercise training and this subsequent PTH increase, it's then going to lead to a progressive breakdown of bone over time. Now, if you know anything about endurance athletes, they tend to train quite a lot. So they'll be doing upwards of 20 plus hours a week. And if you're having periods of extended PTH-induced bone resorption, this can lead to structural changes in bone, which might explain the increased risk factor experienced by endurance athletes. I'd also like to examine the muscle calcium kinetics. So what is it about muscle that releases and uptakes calcium during exercise? And the reason being is, while it seems very sciencey, I suppose, mechanistic in insight, if we can address this issue, then it means that we won't have to have calcium supplementation as a method of reducing this disruption. We might be able to reduce the disruption at the site of the muscle. And then finally, looking at chronic exercise training and structural changes in bone. So I'd like to do some kind of training-based study, having people exercise in one of those 3 different metabolic intensities, and see what the structural changes that occur in bone are. Based on the results, it should suggest that those who exercise between the 2 thresholds would see the greatest loss in bone compared to those who exercise either side of it. Now, I understand that's quite a lot to digest, but if anyone has any questions, please feel free to ask me, or I'm happy to talk about it further down the line.